Welcome to the Concierge Care Health System podcast. The information shared in this podcast is for general informational purposes only and does not constitute medical advice. Listeners should be aware the content is not a substitute for professional medical advice, diagnosis, or treatment. Always consult with a physician or other qualified healthcare provider for medical questions.
Welcome to the Deep Dive. Have you ever paused to think about what the future of medicine truly looks like? We're kind of on the cusp of a transformation so profound, it's shaking the very foundations of how we think about health and a disease. For generations, medicine has often operated on a one-size-fits-all model, but that's changing in a big way right now. Today, we're taking a deep dive into precision medicine. Our mission for this deep dive is pretty clear. We want to unpack what precision medicine truly is, how it actually works, where it's already making an undeniable impact on real lives, the significant hurdles it still faces, and ultimately where this incredible journey is headed. And for this, we've got a stack of fascinating sources that shed light on every facet of this complex and evolving topic.
Yeah, and just to give you the clearest picture right from the start, precision medicine is essentially about moving beyond that one-size-fits-all idea. It's using your unique biological story, your genes, your environment, your lifestyle to tailor everything from prevention to treatment, really making it personal for you.
That's a powerful idea, tailoring medicine just for you. We often hear terms like personalized medicine or individualized medicine. So why did precision medicine become the preferred term within the scientific and policy communities? I mean, it feels like a subtle but important distinction.
It is. It's more than subtle. It was actually a very deliberate and strategic reframing. The term personalized medicine, well, it carried a risk of misinterpretation. It kind of suggested that a unique drug would be designed for every single person. And that's largely unfeasible, right? Both from a manufacturing standpoint and within our existing regulatory frameworks. That misperception could have created a huge barrier to securing the kind of funding and, widespread adoption this field needed. Precision, on the other hand, more accurately describes the actual scientific process. It's about classifying patients into distinct subgroups based on shared disease susceptibility or maybe their predicted response to a particular therapy. This model, stratifying populations into more precise groups, fits much better with how we do clinical trials and how regulatory bodies approve treatments. It was actually pretty instrumental in the successful launch of major national programs like the U.S. Precision Medicine Initiative.
Okay, so it's about finding the right treatment for the right group of people.
Exactly, or subgroups.
Which, it could be a group of one sometimes, but it doesn't necessarily mean a unique treatment for every single person. That makes a lot more sense, practically speaking. And a foundational principle, as you mentioned, is this triad of individuality: genes, environment, and lifestyle. We talk a lot about genetics these days, but why are all three of these crucial?
You're right. Genomics is absolutely foundational. It's our biological blueprint, you could say. But here's the key. It doesn't operate in a vacuum. Think of it like a script, maybe. Your genes are the script, but how that play unfolds depends heavily on the stage and the direction. Environmental exposures, toxins, diet quality, even social factors like where you live. These are critical modulators of disease risk. And then your lifestyle choices, physical activity, smoking, sleep. These are powerful determinants of how that genetic script plays out. And, crucially, how well you respond to treatment. For many common burdensome chronic conditions, things like heart disease, diabetes, most cancers, these non-genetic factors are understood to be just as important, maybe even more so than genes alone. The predictive power of genetics by itself for these complex diseases, it's often surprisingly low to moderate. So you really need that holistic approach, capturing the interplay for precision medicine to deliver on its promise.
That shift in understanding is huge. So if we pivot to the promise of precision medicine, how does it change healthcare's fundamental philosophy? We're not just talking about new drugs here, are we?
Oh, not at all. It signals a fundamental shift in the whole philosophy of healthcare. Moving from mostly reactive, treating disease after symptoms appear, to truly proactive and preventive. Imagine identifying high-risk individuals early, based on their unique profiles. Precision medicine can enable highly targeted preventive strategies, maybe averting illness altogether, long before it even manifests. And for diseases that do develop, it offers the potential to transform treatments by targeting the root molecular cause. That means maximizing efficacy and, really importantly, minimizing side effects. Now, the initial costs can be high, sure, for advanced diagnostics and targeted therapies, but the long-term economic implications, they point towards higher value healthcare, better outcomes, and reducing the overall burden of chronic disease down the line.
That's the dream, isn't it? But this idea of individualized care, treating the patient, not just the disease, that's not exactly a new concept in medicine, is it? Did this have historical roots?
Oh, absolutely. What's fascinating here is that the core idea, treating the patient individually, has incredibly deep roots. Philosophers, physicians, going way back to Hippocrates in ancient Greece, they recognized the importance of individual patient characteristics. Centuries later, Sir William Osler famously said, "It is much more important to know what sort of a patient has a disease than what sort of a disease a patient has." Now, of course, they lacked the molecular basis back then, but they established the underlying principle that individual variability matters.
Okay, so we had the philosophy, and then you mentioned some early scientific glimmers. When did the science really start to catch up? When did we get the tools to look at the building blocks?
Right, that's when molecular biology really entered the picture. The game completely changed with a few key discoveries. First, obviously, the DNA double helix structure by Watson and Crick in 1953. That gave us the blueprint. Then, Sanger sequencing in 1977, which let us actually read that blueprint for the first time. But the real moonshot moment, the biggest catalyst for modern precision medicine, was the Human Genome Project, completed in 2003. It gave us the first comprehensive map of the human genome, an indispensable reference that allowed researchers to start systematically connecting diseases and traits to genetic factors. It really energized the whole field, including pharmacogenomics.
A huge step. But the Human Genome Project, while seen as this ultimate map, it threw a curveball too, didn't it? What was the big, maybe unexpected insight, especially about common diseases?
Yeah, that was a real aha moment. Initially, there was a lot of hope for a simple one gene, one disease model, like a switch for every condition. But as the science matured and we sequenced more genomes, it became super clear that most common conditions, heart disease, diabetes, most cancers, are rarely that simple. They're complex, multifactorial diseases, influenced by the cumulative effects of maybe hundreds, even thousands of small genetic variants, what we call polygenic risk. And those variants are constantly interacting with environment and lifestyle factors. So this realization forced a recalibration. The immediate clinical applications for common diseases weren't as straightforward as hoped. The modern precision medicine movement is really a response to that complexity, understanding we need to look beyond just genetics for these conditions.
Okay, so this recalibration, this new understanding must have led to some really ambitious efforts to actually build the field we see today. Can you tell us about some of those major initiatives?
Absolutely. Things really came together with a major policy milestone in 2015, the launch of the U.S. Precision Medicine Initiative, PMI, by President Obama. This gave it significant federal funding and high-level political backing. It had short-term goals, mostly in oncology, but also a much broader long-term vision. And the cornerstone of that long-term vision is the All of Us research program. It's a massive undertaking led by the NIH. The goal is to enroll at least 1 million diverse volunteers from all across the U.S., and they're collecting not just genetic info, but also tons of data from electronic health records, plus details about environment and lifestyle. It embodies this idea of a learning health system, creating this incredibly rich, diverse data resource designed specifically to tackle those complex, multifactorial diseases.
Wow, 1 million people, that's a massive amount of data. So what are the core technologies, the scientific engines really driving this whole revolution?
Well, what's really powering this is what scientists call the omics cascade. It's like a whole new toolkit for understanding biology at different levels. So you have genomics, that's the DNA blueprint, then epigenomics, which looks at how genes are switched on or off without changing the code itself. Transcriptomics shows the RNA messages being sent. Proteomics looks at the actual proteins doing the work. Metabolomics analyzes the small molecules, the chemical byproducts, and microbiomics studies the microbes living in and on us. But the real breakthrough, the real power, comes from multi-omics integration, pulling all those layers together for a comprehensive, holistic view. It's like building a 3D model instead of just looking at flat blueprints.
That really paints a picture. Let's zoom in on one of those next-generation sequencing, or NGS. What makes it so revolutionary, especially for reading that blueprint?
NGS is basically the cornerstone technology that makes large-scale genomic analysis possible, and importantly, fast and affordable. It's a range of methods that let us sequence millions, even billions, of DNA fragments all at once in parallel. This was a huge leap from older methods in terms of speed and cost. It's why genetic testing is becoming much more common. Its key applications, things like whole genome or whole exome sequencing, which help identify genetic variations linked to rare and common diseases. And in oncology, it's crucial for tumor profiling, analyzing a tumor to find specific actionable mutations that can guide targeted therapies. Of course, all this NGS data is just the raw material. You need incredibly powerful computational tools to make sense of it all.
Okay, so if NGS lets us read the blueprint, then CRISPR-Cas9 is like the gene editing scalpel that lets us actually edit it. That sounds like something out of science fiction. How does it actually work?
It is pretty remarkable, and it actually comes from a natural defense system in bacteria. CRISPR-Cas9 basically has two main parts. There's the Cas9 enzyme, which acts like incredibly precise molecular scissors to cut DNA. And then there's a guide RNA, a molecule engineered in the lab to direct those Cas9 scissors to the exact spot in the genome you want to edit. So the guide RNA Cas9 complex scans the DNA, finds the matching sequence, and Cas9 makes the cut. Once that cut happens, the cell's own repair machinery jumps in. And we can harness that repair process to either knock out a faulty gene, basically disable it, or provide a correct DNA template to knock in a desired sequence. Its relative simplicity, efficiency, and programmability make it a huge leap over older gene editing tools.
The ability to edit DNA so precisely, it's just astounding. Sounds almost too good to be true sometimes. Where is that promise really being explored right now? What are the big therapeutic hopes for CRISPR?
Well, the main promise is its potential to directly correct the root genetic cause of inherited single-gene diseases, monogenic disorders. NGS and AI help find the precise targets, and CRISPR provides the tool to potentially fix them. For instance, clinical trials are looking at correcting the mutation causing sickle cell anemia or beta thalassemia in a patient's own blood stem cells, or editing the faulty CFTR gene in lung cells for cystic fibrosis. There's also immense hope for diseases like Duchenne muscular dystrophy. And beyond those, CRISPR is also revolutionizing cancer immunotherapy. We can engineer a patient's T cells to make them much better at finding and killing cancer cells, like targeting CD19 on leukemia cells.
But that kind of power must come with significant challenges, right? As CRISPR moves more towards the clinic, what are the major hurdles?
That's a really crucial point. Despite the huge potential, there are definitely big hurdles. A major safety concern is off-target, affects the risk that CRISPR might cut DNA somewhere unintended, which could be dangerous. Then there's efficiency, making sure the edit happens correctly in enough cells to actually have a therapeutic effect. That's tough. Delivery is maybe the biggest logistical challenge. How do you get the CRISPR system safely and effectively to the right cells or tissues in the body? People are using modified viruses, tiny lipid nanoparticles, and finally, mosaicism, where only some of the target cells get edited, can lead to unpredictable results. These challenges also create this feedback loop, pushing for even better sequencing to check for these unintended effects.
So all this amazing tech, from omics to gene editing, it generates just vast amounts of data, almost unimaginable amounts. How does the data ecosystem bring it all together and make it useful?
Yeah, the sheer volume of data is staggering. We need a whole new kind of data ecosystem. It has to handle not just big data, but diverse, high-quality, integrated data from all sorts of sources. People often talk about it as a learning system or a knowledge network. It pulls together genomic data, other omics data, clinical info from electronic health records, medical images, lifestyle data from wearables, even environmental data. The ultimate goal is this virtuous circle. Collect large-scale, secure data, use it for research, generate new knowledge and therapies, then translate that back into clinical practice to improve care, capture the outcomes as new data, and feed it back into the system to keep refining it.
That's a really comprehensive vision, but let's get practical. Where has precision medicine made its most significant impact clinically right now, today? Where can we actually see it changing lives?
Without a doubt, oncology. Cancer treatment, that's really the vanguard. Precision medicine has fundamentally changed how we classify and treat cancer. We're moving beyond just defining it by where it is in the body. Now, cancers are increasingly defined by their underlying molecular changes, and this is possible because of routine biomarker testing, analyzing a patient's tumor, often with NGS, to find that specific genetic or protein changes driving the cancer. Those findings then directly guide treatment choices.
Can you give us some landmark examples, stories of real breakthrough cancer treatments?
Sure. A huge success story is imatinib, brand name Gleevec, for chronic myeloid leukemia, or CML. CML is driven by a specific genetic glitch, the BCR-ABL fusion gene. Imatinib was designed to specifically block the overactive protein made by that gene. It transformed CML from what was often a rapidly fatal disease into a manageable chronic condition for many people. We've seen similar successes in solid tumors too, like trastuzumab or Herceptin for HER2-positive breast cancer. It targets the HER2 protein. And in non-small cell lung cancer, finding specific driver mutations in genes like EGFR or ALK led to highly effective oral drugs, tyrosine kinase inhibitors that have dramatically improved outcomes. They're standard care now.
And what about these living drugs we sometimes hear about in cancer treatment? They sound pretty revolutionary.
Ah, yes. That brings us to CAR T-cell therapy, a prime example of a living drug. It involves taking a patient's own T-cells, which are immune cells, and genetically engineering them in the lab. They're engineered to express a receptor that specifically targets a protein on their cancer cells, like CD19 on leukemia cells. Then these supercharged cells are infused back into the patient. They act like a potent, personalized army against the cancer. The story of Emily Whitehead, the first child treated with CAR T for acute lymphoblastic leukemia who had this incredible lasting remission that became a really powerful symbol of this approach.
Wow. Okay, moving beyond cancer, how is pharmacogenomics tailoring drug therapy? It sounds like it could prevent a lot of that frustrating trial and error with medications.
Exactly. Pharmacogenomics is a critical piece of precision medicine. It focuses on how your unique genetic makeup influences how you respond to drugs. The goal is just what you said. Use genetic info to prescribe meds that are safer and more effective, getting away from trial and error. A classic example is the HIV drug abacavir. About 5–8% of people used to have this severe, sometimes fatal, hypersensitivity reaction. Research found it was almost exclusively in patients with a specific genetic variant, HLA-B57.01. So now the FDA recommends screening for this allele before starting abacavir. It's virtually eliminated that dangerous reaction. Another well-known use is guiding the dosing of warfarin, the blood thinner. Genetic variants in CYP-C9 and VKORC1 can explain a lot of why people need different doses. Genotype-guided dosing helps reduce risks of bleeding or clotting.
That's genuinely life-saving. What about the thousands of rare genetic disorders? Is there new hope for conditions that were often considered untreatable?
Yes, definitely. For thousands of rare diseases, most caused by a single faulty gene, precision medicine offers a totally new kind of hope. The aim is to correct the underlying defect, potentially offering a cure. Cystic fibrosis, CF, is a fantastic example. It's caused by mutations in the CFTR gene. New drugs called CFTR modulators, like ivacaftor and Trikafta, target specific mutations. For patients who are eligible, these drugs have truly transformed CF from a fatal childhood disease into something manageable in adulthood. Huge improvements in lung function and quality of life. Similarly, for spinal muscular atrophy, SMA, a devastating neuromuscular disorder, there is a gene replacement therapy, Zolgensma. It delivers a working copy of the SMN1 gene. For infants treated early, this one-time infusion can be absolutely life-saving.
These single gene disorders are seeing amazing progress. But what does this all mean for the common multifactorial diseases like heart disease or diabetes? They're not caused by just one faulty gene. How does precision medicine tackle those?
You've hit on one of the biggest ongoing challenges. These conditions are way more complex. It's this intricate interplay of maybe hundreds or thousands of genetic variants, each with a tiny effect, combined with a lifetime of environmental and lifestyle factors. So the approach isn't about fixing one gene. It's more about using comprehensive multi-omics data and powerful statistical tools like polygenic risk scores, PRS. Think of a PRS like this. Instead of one big on-off switch for a disease, you have thousands of tiny dimmer switches. Each one only shifts the light a little bit. But the combination of how all those dimmers are set gives us a much clearer picture of your overall risk profile, seeing the whole orchestra, not just one instrument. By grouping patients based on these molecular profiles or risk scores, clinicians can get much better at predicting who's likely to get a disease and which subgroups might respond best to certain interventions. It really highlights this hierarchy of tractability, faster progress in diseases with strong, clear molecular drivers, and a slower, different kind of progress for complex ones focused more on risk stratification and matching treatments probabilistically.
Right. That explosion of data from all these technologies seems like it ties directly into artificial intelligence, AI. How is AI helping here? Is it just crunching numbers?
It's doing way more than just crunching numbers. AI, especially machine learning, is really the indispensable brain needed here. It processes these vast, complex data sets, finds subtle patterns humans might miss, and helps translate it all into actionable clinical insights. What's unique about AI is its ability to integrate all these diverse data types, genomic, clinical, imaging, lifestyle, into one analytical framework. It gives us that holistic view, seeing the whole forest, not just the trees.
So are there different kinds of machine learning that are particularly helpful for different jobs in precision medicine?
Yes, absolutely. Different tools for different tasks. For instance, unsupervised learning is great for discovery. It can identify completely new disease subtypes or group patients based just on their molecular data without needing pre-existing labels. Then there's supervised learning. This is trained on data where we already know the outcomes. It learns relationships to make predictions like disease risk, prognosis, or how someone might respond to a drug. And reinforcement learning is interesting too. It learns by trial and error, essentially, making decisions to achieve a goal. It has huge potential for optimizing treatment plans over time, adapting as a patient's condition changes.
Where is AI making a difference in diagnostics right now? How's it helping doctors?
AI, particularly deep learning and computer vision, is really boosting pathology and radiology. In digital pathology, AI algorithms can analyze high-res scans of biopsies to automatically spot cancer cells, grade tumors, quantify immune cells. It speeds things up and can improve accuracy. Then there's radiomics. AI extracts quantitative features from scans like CTs or MRIs, features humans might not even see to predict things like a tumor's molecular subtype or how aggressive it might be, sometimes just from the scan. And for risk stratification, AI models can combine electronic health records, demographics, and other data to produce really accurate risk scores for various conditions, sometimes even outperforming traditional methods.
That's fascinating. And how is AI speeding up the development of new treatments?
It's making an impact across the whole R&D pipeline. For target identification, AI can sift through massive biological databases way faster than humans to prioritize the most promising drug targets. Saves huge amounts of time and money. In drug design, generative AI models can actually design novel drug compounds, and predictive AI can virtually screen millions of chemicals for therapeutic potential, cuts down on costly lab work. And for clinical trial optimization, AI can analyze patient data to find and recruit the best candidates for a trial much faster. This speeds up trials, reduces costs, and boosts the chances of success.
Incredibly powerful stuff. But with all that power, what's a major hurdle for actually getting AI into routine clinical practice?
Well, a primary barrier is often called the black box problem. As these AI models get more complex to handle all this multi-omics data and make sophisticated predictions, their internal reasoning can become really opaque, hard to understand how they reached a conclusion. And clinicians, regulators, they're understandably hesitant to trust a medical recommendation if they can't clearly see the reasoning behind it. This lack of transparency is a big hurdle for adoption and approval. It's why there's so much focus now on developing explainable AI, XAI AI, that can actually explain its reasoning in human terms.
Okay, so the science is incredible, the tech is advancing. But let's shift gears a bit. What are the significant challenges facing precision medicine beyond the science? What are we up against in actually bringing this to everyone?
Yeah, these barriers aren't isolated issues. They're really a complex, interconnected web. Scientific, economic, logistical, ethical, legal, social, they all play a part. Getting to widespread, equitable implementation needs a really sober assessment and a concerted effort from everyone involved. It touches so many parts of our healthcare system and society.
What are some of the scientific or technical hurdles related to just the biology itself that still keep researchers up at night?
Well, in cancer, tumor heterogeneity is a big one. A single biopsy might only capture a fraction of the tumor's genetic diversity. You might miss subclones driving resistance. There's also a huge need for better biomarker validation. We need to reliably distinguish the driver mutations that are actually causing the cancer from the passenger mutations that are just along for the ride. And we often lack good tools to monitor treatment response and detect resistance early on. Finally, data integration and quality are massive technical challenges. Electronic health records are often messy, unstructured, built more for billing than research. Combining that reliably with clean omics data across different places is really hard.
And beyond the science, what about the economic and logistical side? These therapies sound expensive.
They often are. The high cost of therapies and tests is probably the most immediate obstacle. Sequencing costs have dropped dramatically, which is great, but the targeted drugs and especially gene therapies, they can be incredibly expensive, sometimes millions of dollars. That raises huge questions about affordability and sustainability for healthcare systems. And that leads directly to reimbursement uncertainty. Payers, like insurance companies, understandably want strong evidence of clinical utility and cost effectiveness before they agree to cover these new approaches. Generating that evidence quickly can be tough. Beyond cost, there are major infrastructure deficiencies. Most hospitals and clinics just don't have the IT systems needed to handle and analyze these massive data sets securely. And critically, there's a lack of a properly trained workforce. Most doctors, nurses, pharmacists haven't been trained in genomics, bioinformatics, data science. They need that fluency to interpret these complex reports and use them effectively.
These are clearly huge system-level challenges. But let's talk about some of the deeper societal concerns, too. Data privacy, for instance. With all this incredibly personal information, what are the risks?
That's paramount. Genomic data is arguably the most personal information there is. It holds clues not just about your health, but potentially your relative's health, too, protecting it from unauthorized access, cyber attacks, misuse. It's a massive ethical and legal obligation. The large, centralized databases needed for research are essential, but they also become attractive targets. So we need really robust encryption, strict access controls, and clear rules, like adhering to regulations like GDPR and IPAW, to build and maintain public trust. Without trust, people won't participate.
And the fear of genetic discrimination, how's that addressed? Or maybe, how is it not fully addressed right now? It's a real worry for people thinking about these tests.
It is. It's a significant public fear that genetic information could be used against you in getting a job or insurance. And that fear can stop people from getting tested or joining research. In the U.S., the main law is the Genetic Information Non-Discrimination Act of 2008, GINA. GINA does prohibit health insurers from using genetic info for eligibility or premiums, and it stops employers using it for hiring or firing. That's good. But and this is a really crucial, but GINA has major limitations. It does not apply to life insurance, disability insurance, or long-term care insurance. So an insurer could legally require genetic testing or use your existing genetic information to deny you coverage or charge you more for those specific products. It also doesn't protect people who already have symptoms or apply to very small employers or the U.S. military. These gaps leave a lot of people potentially vulnerable.
That's a vital distinction for people to understand. Thinking about these gaps, another huge concern is how precision medicine could accidentally make existing health disparities worse, not better. What's the risk there?
This is a profound and really pressing challenge. There is a serious risk here. Genomic databases, which are the foundation for discovering disease links and training AI, are overwhelmingly based on data from people of European ancestry. As a result, the genetic tests, the risk scores, the AI tools developed using that data might be less accurate, less useful for people from underrepresented racial and ethnic groups. That could lead to major inequities in care. Plus, the high costs of these technologies and the fact that expertise is often concentrated in big academic centers creates access barriers for rural communities, low-income groups, minority populations. It creates this dangerous feedback loop. Lack of access leads to underrepresentation in research, which leads to biased tools that don't work as well for those same populations. We absolutely have to tackle this head-on.
That's a powerful point. It highlights the ethical need to get this right. So, looking ahead, what's the ultimate vision? What does this integrated, data-driven, preventive future actually look like, say, a decade or two from now?
Well, the ultimate vision is a deeply integrated, data-driven, preventive, and truly personalized healthcare ecosystem. One that fundamentally shifts the focus from reacting to sickness to proactively managing health. Imagine a system that knows you really, really well, biologically speaking.
And tell us about digital twins. I've heard that phrase sounds futuristic. How do they fit in?
Digital twins are basically dynamic, computational models of an individual patient, not static snapshots, but constantly updated. They'd integrate multi-omics data, electronic health records, imaging, and crucially, real-time data from wearables, tracking activity, glucose, sleep, you name it. AI would power these twins, integrating all this diverse data to simulate how a disease might progress for you, specifically. And you could virtually test thousands of potential treatments or lifestyle changes in the model before recommending them to the actual person. It represents the ultimate in personalized treatment planning, really optimizing care at the individual level.
That level of personalization is incredible. You mentioned that fundamental shift from reaction to prevention earlier. How does this future vision really amplify that?
Yeah, that's a core goal. Changing healthcare from reacting to disease to proactively preventing it. By using genomic info, especially those polygenic risk scores we talked about, combined with lifestyle and environmental data crunched by AI, it should become possible to spot individuals at high risk for common chronic diseases, cancer, type 2 diabetes, heart disease, long before symptoms appear. This early identification allows for highly personalized prevention plans. Maybe a tailored diet, more frequent screening, maybe early targeted medications. The potential is to dramatically reduce how often these diseases occur, improving population health and easing the huge financial strain on healthcare systems.
So bring it all together for us, the learning healthcare system.
Yeah.
What does that look like when all of these pieces, the tech, the data, the AI, the personalized prevention, finally come together?
The learning healthcare system is kind of the ultimate synthesis. It's where the line between clinical care and research really blurs. Every patient interaction, every test, every treatment, every outcome generates data. This data, with the patient's explicit consent, gets securely, ethically de-identified and fed into a massive interconnected knowledge network. AI continuously analyzes this data stream to find new biological insights, validate treatments, generate new hypotheses, and then this new knowledge gets fed back almost in real time to doctors and patients at the point of care. It creates this rapid, continuous learning loop. It also implies a changing role for the physician, right? Maybe less the sole expert and more a clinical data navigator or systems interpreter, guiding patients through these complex insights, engaging in shared decision-making for truly personalized care.
What a journey we've been on today. Precision medicine is clearly a profound paradigm shift. It's way more than just an incremental improvement. The progress so far is undeniable, isn't it? Transformative results, especially in cancer and rare genetic disorders, those landmark therapies, they're powerful proof of what's possible.
Absolutely. And while the science and tech keep accelerating, opening new doors, the path to making this widespread and equitable is definitely challenging. The scientific hurdles are still formidable, understanding tumor complexity, applying this to common diseases. The economics, high costs, reimbursement issues create real access barriers, but I think most critically, those ethical, legal, and social implications, data privacy, genetic discrimination, the risk of worsening health disparities, they demand our most careful attention. The gaps in our legal protections, the biases in our data, the inequities in our healthcare systems, honestly, those might be the biggest obstacles to realizing the full positive potential of this new era.
So fulfilling that potential really demands a concerted, collaborative effort, doesn't it? From everyone involved. Scientists need to build diverse data sets. Healthcare systems and educators need to invest in infrastructure and training. Policymakers need to create robust frameworks that protect people but still foster innovation. And all of us as a society need to have an open, ongoing conversation about the values that should guide how we use these powerful new tools. The journey toward precision medicine is definitely a marathon, not a sprint. But it leaves us with this thought. What will it truly mean for your health and for our entire healthcare system when medicine isn't just precise, but becomes a truly continuously learning system? And how will your unique data contribute to the health of tomorrow?
Concierge Care, providing world-class, personalized care for patients and members. Taking a proactive approach by offering its free digital health clinic for anyone in need of these services. Reach out to us via our website, www.health.conciergecare.net, or give us a phone call at 786-809-2345.